See how I said I only have 5 minutes? Okay, you are in note video 2. So what I was saying, you've got 2 alleles for each trait that are separating during meiosis. This is anaphase 2. Those bits are pulling apart. During fertilization of your egg and your sperm, those 2 alleles are going to come back together because remember, eggs have 23 chromosomes, sperm have 23 chromosomes. You need 46. You need 2 of each. Heterozygous organisms are called hybrids. It's a hybrid. You have one capital, one lowercase, one dominant, one recessive. You have one of each. It's a mixture. They are hybrids. Remember my explanation of hybrids when, like mules, you have a horse and a donkey. You have one of each. They cross and they make a mule. A mule is a hybrid. It has half of its genetics from one type of animal, a horse, and one half of its genetics from the other type, a donkey. This is the law of independent assortment. Big words. Promise it'll make sense. Random distribution of alleles occurs during gamete formation. So when, remember we've got crossing over on those chromosomes. So the crossing over chromosomes, they swap genetics. This is how we get things that are completely different. This is why your siblings look different. Gametes on separate chromosomes sort independently during meiosis, crossing over. Each allele combination is equally likely to occur. So here is this allele, allele number 1, allele number 2, number 3, and number 4. These are all different alleles. Remember, you have to have 2 alleles for your genes. These are a possible combination of what you can have. Each one has an equal representation, a 1/4 or a 25% chance of being passed on to the next generation. This is called a monohybrid cross. So looking at the image, you see one letter. We're looking at the letter Y. This is the only kind of cross we're worried about. So remember the prefix mono means 1. So this diagram shows how Mendel continued his experiments by allowing the heterozygous yellow plants, so capital Y, little y, to self-fertilize. So right here, this is the one he's talking about. This is the F1 generation. This is a monohybrid cross. You have one type of allele that you're looking, or one genotype you're looking at, capital Y, lowercase y, crossing with the exact same genetic type. Mono 1, so one type, we're looking at Y, and it's a hybrid. We have a capital and a lowercase. That's your hybrid, one of each. So it's one hybrid crossing together to get all this stuff. So we have the possibility of getting 3 different genotypes. We have homozygous dominant, heterozygous, same thing, and homozygous recessive. So a cross that involves hybrids for a single trait, like your Ys, are called a monohybrid cross. 1. So looking at our Punnett squares, we've already started practicing this, but we didn't get that far into it. Punnett squares are used to predict the possible offspring of a cross between 2 known genotypes. I want you to do this right now. See how she's rolling her tongue? I want you to see if you can do it. If you can, it's kind of hard to see with this little hidden thing, but if you can, you have the dominant trait for tongue rolling, if you can make that shape. If you cannot, you are recessive. If you can do it, that means your genotype can either be homozygous dominant or heterozygous. If you cannot do it, this is your genotype. So I can do it. So I either have this or this genotype. So this capital T, it's dominant ability to roll the tongue. Remember, you only need one to be able to do it, to be able to have the phenotype or what you see. So we talked about a monohybrid cross. That was 1. Let's talk about a dihybrid cross. The prefix di means 2. 2 hybrid crosses. This means the simultaneous, or at the same time, inheritance of 2 or more traits in the same plant is a dihybrid cross.